All right, as a few more people are coming in and getting settled, I think I'll start my introduction. So we are super excited today to have one of our own to give our distinguished seminar series lecture, and that's Dr. Valentina Greco. Valentina originally hails from Palermo, Italy, and carried out her PhD in Germany at the EMBL and MPI, and then moved to the Rockefeller University for her postdoc, where we were both postdocs in kind of an overlapping fashion. So particularly super fun and very high energy place to do one's postdoc. And Valentina took that energy and brought that, I would say, here to you. She is now the Carolyn Walch Slayman Professor of Genetics. And for those of you who knew Carolyn, like I did, I think that's a particularly special honor for which Valentina is very well deserving. Because, Carolyn was a real proponent of really good science, had fantastic taste in basic science with the potential for impact. And I think that really fits so well with Valentina's work. So in addition to being a member, of course, of our cancer center, Valentina's primary appointment is in genetics, and then she holds secondary appointments in cell biology and dermatology. And she's also a member of the Yale Stem Cell Center. And she was very recently, among many other honors, named a Howard Hughes Medical Institute investigator. So what brings her here today to the Cancer Center? I think as a cell biologist myself, I think one of the most exciting kind of emerging themes, which Valentina has really been a pioneer in, is understanding how the normal homeostatic mechanisms in tissues can play important roles in combating cellular transformation and tumorigenesis, right? So the idea being that the tissue is able to surveil itself and to be able to identify and then act on cells which are no longer behaving in the appropriate fashion. And so unlike our kind of classic, P53 tumor suppressors, this kind of model working at the tissue level is really exciting. And in order to make those discoveries, Valentina has been a pioneer in live cell vital microscopy. So I'm sure we're going to be treated to movies such as this. We were just talking as we were starting, as someone, again, who's a cell biologist who loves these kind of movies. It turns out that actually it's very challenging to take really great movies and have the right instruments to do that. But actually then analyzing this data is also a really big challenge and something that requires constant innovation to really get down to the mechanistic details we can. And so I'm very excited for the talk. Thank you, Jennifer. You can clap. You can clap. Start. Oh, yes, I'm actually. So one of the things we love about this, I love about this series is that I'm able to give Valentina this really nice,
Thank you so much.
Gift to memorialize today's lecture. And now I'll get out of your way.
No. Thank you so much. All right. Always a lot of excitement and anxiety at the same time, speaking to people that I know and I care about. And also, as I was putting together the talk, that inevitable choice that I'm confronted with to not be able to talk about the projects. And so this is an expression of love for people here in the second row from my lab for whose project I wasn't able to represent, even though I care deeply about. So the invitation to speak in a distinguished speaker series comes for me with both the honor, but also the responsibility to open spaces for conversation that we don't have or we don't sustain as much. I start by saying that we divorce science from identities. And yet in what my experience and the experience of people I've been interacting with has been that lived experiences are a powerful force for us to start to create out the lenses with which we do scientific discoveries. What I mean by that is we are influenced by not only the membership we belong to, but the experiences we have made to ask questions, but also to not ask certain questions. And so that deeply influences the research we do. And so in this talk, you're going to see 4 parts. The first one is how we in the lab view scientific discoveries and the importance of those lenses. Then how those lenses have allowed us to interrogate regeneration using the skin of live mice. The third, the contribution that these lenses have allowed in the epithelial stem cell field. And lastly, where we are going next. So we pause and think a lot in the lab of what creates scientific discoveries. And I think in society, there is a narration that supports the individual level interpretation. So there are brilliant individual insights, so even serendipitous events. But we don't ever speak about the other side of it, which is not only individual, but also the collective and the power of groups. For instance, if you take what the lab has been able to do is bridging 2 fields that didn't meet before. Developmental biology has been used live microscopy for a long time, but mammalian regeneration had not. And so it is 2 fields that come together that created the capacity to then make contributions, as well as the very important and vibrant intra and interlab interactions. And so when thinking about this question and the thrill I feel in partnering with scientists in my lab and beyond is thinking about both my lens and my role. With respect to my lens, I think it's easy for us to interface with each other when we do research and have this lens of a scientist, but it's less easy and not enough spaces are there to think about other dimensions of our identity. For instance, when I reflect on myself, I think about my identity as a cis woman, as an immigrant, and as a mother. Why do they matter? With trepidation, I decided to give you a couple of quotes since I feel very celebrated and loved in this school by telling you that it still exists and it still persists. So I'm giving you just a couple of quotes, for instance, like this one, where at the poster still recently, when asking a question to a male trainee, the male trainee still turned to the male besides me rather than me, even though I'm the one asking the question. In addition to be a repeated event, it creates a small weight that one carries. Not too long ago, a senior faculty approached me saying he was worried about my career. Well, from a standard perspective, the CV was already full of those shining marks that we think the profession recognizes as important. And this person asked me to consider taking classes to correct my accent. The counterpart male that was considered for a chair position and had an accent wasn't advised the same. And bad news for all of you, I didn't take those classes. When I was winning an award along with 4 other colleagues, 2 females and 3 males, we were each interviewed for half an hour. And so this journalist had collected enough information from each of us. And yet the final article had 1 quote from me, no quote from the other female, a collective 7 quotes from the rest of the males. This list goes on and on and on. And the more I was writing just to think about this, the longer the list became. By any means, I don't want you to feel sorry for me, maybe empathetic if you have experienced it, but trying to create a space where we don't understand the experiences of others, and we don't understand the forces in which our professional society imposes on all of us. But this collective burden creates a large weight, which made me feel really excited when I saw this work of Susan Clinard. Is a native from Chicago, resident here in New Haven. The series called The Way We Carry. You can see the body of this person deforming and bended, and the way that all this continuous aggression can create on the body of somebody. Now, if I take this and put now the spotlight on me and hold the mirror to myself, I have many other membership, many other identities, such as white and able, where I'm on the other side of this privilege. So unless I become myself curious about understanding the experiences of others without really having experienced it, I don't think we can move forward as a profession. And so the perspective is that each of these can be a source of insight, creativity, and contribution, but it demands that this multitude of membership are understood in the context of the groups that we lead. So all members in our lab have all these different identities. And so my role has been to figure out how to create an environment that can unlock the creativity of each member by creating processes and structure that can connect within and between membership. So a number of principles that I've been driving since the last 15 years, my lab has been committing to be curious and open to the experience of others, as well as have a regular discussion about futures of the lab members. And also working on implementing feedback on me. Without a third party creating anonymous feedback on me, I would not be able to see what is difficult for me to see. And as well as understanding that we are not hired to be manager and yet we manage groups. And so the importance of courses and training for us to continue to innovate and keep current the capacity to manage groups. And so with these lenses in mind in these processes, what the lab and I are fascinated about is understanding this process, which is highly dynamic. We lose and replace 50 to 70 billion of cells daily. And this is true across a number of tissue, blood, skin, intestine, where we can ask fundamental basic questions that many are interested in. For instance, do all stem cells in a population have the same potential, or is there a hierarchy? Also, how the environment and neighboring interaction are influences those choices. And finally, how stem cell coordination contribute to tissue architecture and function. So when thinking about this question, we leverage the skin not only because of a prior expertise, but also that I had, but also because of the unique accessibility of microscopy to this organ. And this is building not only on decades of work on microscopy done in developmental biology. In the regeneration field, prior to us, Yoshida had been able to create advances in testis stem cells, Cristina Losalso in bone marrow. And also there was a wealth of knowledge that was coming from the skin field to understand molecular mechanism of this regeneration occurring in the skin. And lastly, and critically for me and the lab, is that when I started, I was very lucky to be approached by Anna Haberman and Dave Gonzalez, who run this attack intravital facility. Professor Haberman was really kind to give me a couple of sessions for free at the 2 Photon just to play around. And that's how we together figured out that there was an opportunity to use this type of microscopy to study the skin epithelium. And so what you're seeing here is a plunging into the skin from the surface where you touch when you touch your skin all the way down to the muscle, where you're seeing in different color, different cell types, and provides here an opportunity of understanding homeostasis within different tissue types, the characteristic of those interactions, and then also what pathway regulate all these interactions. And what the lab has done is focusing initially in this layer, which is the basal layer. If I give you schematics, here is where you touch your skin, all these differentiated layers, all the way down to the stem cell layer that you're seeing now in an XY view, mirror image what you're seeing here, which is a collection of nuclei in the basal epithelial stem cell layer. And if you color, for instance, membrane and nuclei, and you watch it over time, you can start to ask questions such as who proliferates, when they proliferate, what are the neighbors doing, and what are the consequences at the tissue level for this activity. So in leveraging the passion that we feel for movement, color, and shape, a major question that the lab has been starting to approach is understanding behavior, cellular state, and molecular state. And this is the tremendous work of several in the past years that I'm going to highlight over time. So beginning with behaviors, if you take a top-down view of the basal layer, all these cells perform two behaviors. One is division in blue, and the other is delamination by differentiation. Practically, what do I mean by that? If you take an XZ view, cells that are residing in this basal layer in orange are going to delaminate after they've been differentiated, which means they're going to go and be integrated in upper layers, creating the barrier of the stratified skin epidermis. And so what we had previously shown is that some cells have an equal potential to perform these behaviors. But what we didn't know is what is the scale of this coordination? Is it occurring at the tissue-wide level, or is it local? And also the temporal order of behaviors. Prior research was hinting that the proliferation may be a driver for the cells then to leave by delamination. And so in order to address this question, there had to be a couple of major innovations. One, Kai and Katie team up to be able to go to the same here cells within the stem cell layer, these are nuclei, over 12 hours for 7 days in a row. Then we had to be able not only to segment, but track every single cell over time. And it would not have been possible if we didn't team up with Alon Klein and Kyogo Kawaguchi, who had the statistical competence and power to be able to extract the information. What you're seeing at the beginning of this movie is that everyone has a number, and over time, the cells that have a number are cells that are dividing. There is so much activity that it's very difficult to manually understand when a cell is performing the behavior, what are neighbors of first degree and second degree doing, either after or before the event. And so without the statistical power, we wouldn't have been able to understand when we analyze all this data, that if you take this as an inset, a representative inset, the cells with an asterisk are cells that are leaving this plane and going up to differentiate, that within 24 hours, neighbors would expand in size, and then one neighbor would divide to compensate for that cell leaving. So this was exciting to us because it reversed the order with which we expected things to happen. A cell is creating a loss of space because it's leaving, and therefore the neighbor is compensating by proliferation. And so when thinking about in a cartoon manner how we understand these two behaviors, what we understand is that there is a local level of regulation where coordination of behavior is at the level of direct neighbors, and that delamination drives proliferation. Now, this is different when we think about embryonic stages, where instead what we learn from Sarah Wikström's work is that as the tissue needs to build and be created, then proliferation will drive differentiation. Instead, what we are looking at is an adult stage when the tissue needs to be simply maintained, in which then is the loss of a cell that will trigger proliferation. So what we wanted to do next is understanding how this delamination is driven, since the differentiation is upstream of that, and it required visualizing the cellular state of differentiation. So we had to team up again with other people that had competencies we didn't have. In this case, it's Maria Kasper and Carla Nussberg. They were at the time also pioneering the skin field, the capacity to look at the single cell resolution, the transcriptional profile, but also understand through a series of markers where cells reside. For instance, all the cells here up until this dashed line that are in the basal layer I've shown you before, and past this dashed line, the cells have left the basal stem cell compartment. What they saw is that as stemness genes, for instance, keratin 14 is representative of that group, they were down-regulating while the cells were still on the basal layer. Differentiation genes were ramping up. This is true for multiple differentiation genes, and they notice a continuum and a way in which various differentiation genes were being upregulated. The good, what we liked very much is that the keratin 10 was the first one ramping up, because coincidentally, a colleague, Terry Leckler, had developed a reporter that allows us now to combine single RNA-seq analysis to live imaging. So what you're seeing here is a field of view where each, in pink, you see the membrane of all these cells in the basal layer, and in green, you're seeing a reporter that tells you when the cells are turning on the differentiation genes. And so what Katie then could do is to track the same cells over time, and in fact, retrospectively too, because after collection of data, you can see the cells that have left when they have turned on that reporter and when they were initially negative for it. This is a very powerful approach to understand two things. One is how long do cells turn on that reporter and still stay in the basal layer, and then when that will lead to an event of an exit from that layer. And so the behavior combined to the cellular state. What was really exciting is Katie discovered that while these cells were sitting on the basal layer, so already differentiated and committed to live within 4 days, they could still proliferate. That was different from the way the field was thinking, because it makes compatible to have two different behaviors, differentiation and proliferation. And what Katie discovered is that this is a need of the tissue to maintain basal cell density. So let me summarize what I told you so far. So from single RNA sequencing data from multiple labs, we interpreted the heterogeneity within the basal layer as the existence of multiple cell types or multiple subpopulation of stem cells. What we think we have understood is that all these data are also compatible with the model where there is a single population of stem cells, where they are simply transitioning through differentiation state by evolving their gene expression. And there is a process that takes days as to allow also for proliferation to be possible and to maintain basal cell density. So this type of insight allows us then to continue our quest in understanding now at the level of transcription, how we understand the regulation of critical genes like keratin 14, which is the stemness genes, as well as keratin 10, which is this transcription differentiation gene regulator. And you're required to visualize their transcriptional regulation. So a new adventure in the lab has been to start to understand through an MS2 system, which is a system that allows us to see the endogenous gene being transcribed. And in this case, if you look at this picture, you're seeing the nuclei all in pink. And when you see this foci, you understand that the transcription of this particular gene, in this case, is on, as well as the transcript distribution within the cells. This was an exciting area that was contributed by Danny May and Samwon Yoon that allows us now to understand all the way down to a molecular tracking what are the decisions behind the behaviors we study. And what really fascinates us, and this is a video from Jason Lin, is that there are ways to look at gene expression that come from a developmental biology perspective. What do I mean by that? In these very preliminary videos, Jason may think that some of the cells may sync in their transcription activity. We are going to go and continue to do this type of analysis. But what this brings up is a lens, which is a neighboring lens that is not as present in gene expression, but so present in developmental biology that can inform us in the way we are going to look at the data. So if neighborhoods are present, then how is this synchronization regulated? Does it continue and sustain throughout lifetime? Is a property of a specific transcript or is a shared one, for instance, housekeeping genes and other genes? So this is just to give a window into the opportunity the combining field can continue to give. So if I go back and summarize what I told you so far is that at the neighborhood level, the coordination that we discover is across scales, behavioral all the way to molecular. And manipulation, I haven't time to show you, show that this coordination is critical for tissue structure and function. Also, the flexibility is rooted in the temporal scale. So not necessarily the cells themselves, which of course have the capacity to be flexible, but the process itself is slow enough that allows for that for that flexibility to be rooted, so that this layer is a degree of differentiation states that can all participate to proliferation, but be ready to be moving up to form the barrier. So when thinking about this coordination, the last bit on this epithelial stem cell regulation is understanding how this coordination is achieved tissue-wide. And this has been our efforts in the signaling area. You will see a bit later Tianqi Qin work, and in this case, Jess Moore, who was turning into calcium because of the role that we knew had already played in the differentiation of the skin epidermis. But what she was really surprised by is to see all this incredible activity that calcium is having on the basal layer of these cells, even though these cells are non-excitatory. And when we were looking at it, even though Jess had already evolved a number of computational approaches, it initially felt almost quite random. And it wasn't until we partnered with Smita Krishnaswamy and Dhananjay Bhaskar that we were able to develop machine learning approaches that allow us to make sense of this calcium. And then the connectivity that then just discovered of all this activity, the tissue-wide level, and how it differs actually when we look the same comparison with other tissues, such as neural visual cortex. This is a very fast vignette, but just to tell you that once again, with teaming up with other expertise, that we are capable of making more sense and go deeper into quantification of all the data that we get from the live imaging. Okay, so far what I've told you is our excitement about mechanisms that endow regeneration over time, and the need and the pleasure of teaming up with so many other expertise in campus or elsewhere in order for us to tackle the complexity of stem cell regeneration. And there are two major themes that I'm going to talk about now and give you a few vignettes for each one of them. The first is understanding this homeostatic mechanism in the context of genetic mosaicism. So we all know that since several decades, and also with our own dark brush that show that for P53, that our tissue are full of mutation that we coexist with and we co-evolve with. So understanding this process has been an area of research in the lab. And we started from the early days of the lab all the way to now, where we have started to tackle this question by initially looking at beta-catenin oncogenic mutation. And so when looking at that particular mutation, for us was a way to enter into a system that altered the tissue architecture and that led from the field to tumors that are quite benign. They are called polymatricoma. And what the field understood is as you express this activated beta-catenin within the skin epithelium, you might already notice that these two mice look different. There is much more fur and there is more skin in this mouse, that you form these tumors, and that was the state of the art. But when Samara and Christy team up, they started to ask how do healthy tissue, harbor this mutation, and how is architecture deformed? So what you're seeing here is the capacity of looking at a mouse that is anesthetized, and then you're seeing all these follicles here, where each individual dot is a nuclei by fusing an H2B with GFP. And you're seeing the stereotypic organization architecture of the follicle. Then when Samara and Christy induce a mutation through a system that is inducible, so it has a promoter that allows you to be confined within the epithelium, and a tamoxifen regulatable element that allows you to decide when you induce that mutation, you can completely change the field of view. But if it wasn't for Samara, they started to realize that there was something interesting happening, that we would not have finalized our discovery, that all these tumors, when you follow them over time, they're actually gone. These are the same areas in the same mice that show the incredible power that the skin epithelium has to recognize and correct aberrancy and go back to what looks like a homeostatic state. So this was extremely exciting and created a new area in the lab to start to understand essentially the mechanism behind that. We know at the epithelial tissue level there is this clearance, but what are the cellular mechanisms that mediate regression and the underlying molecular mechanism? And so this is a video from Elizabeth Deschênes-Jacobs and Peggy Myung, who is also here a faculty in pathology. And what they were seeing is that if you look at this follicle here, this is the normal axis or the healthy axis of the follicle. These tumors started with this hunchback in many of those follicles. And even though it's only an approximation, the red is an approximation for the mutants, it has allowed us to start to have some insights that this growth were a composition of both mutant and wild-type cells. So these are two separate alleles, so you recombine the oncogenic mutation separately from the fluorescent reporter. But we consistently saw that there was this mixed population that was contributing to this growth. And what Peggy and Elizabeth realized is that also that through an analysis of the Wnt pathway, these beta-catenin mutant cells upregulate Wnt production through a regulator called Wintless. And that, in turn, allows for the wild-type cells to amplify their number. Initially, we interpreted this as to say the wild-type cells had been corrupted, and that was the framing with which we understood that. But then as Samara and Christy understood that these growth, which are, again, are a mix of mutant and wild-type cells regress, we understood that there was more to this story. And so what Samara and Christy started to want to ask is, what is the role of wild-type cells? And it fascinated us because often we stay focused on the tumor per se, on the mutant cells per se, and we ask a bit less, but more in developmental biology, what is the impact of all the contexts, and especially wild-type cells? And so what is the relationship between these two cells? And what, building on Peggy and Elizabeth's work, Samara and Christy decided that they could both slow down the proliferation of wild-type cells by acting on the signaling pathway I was telling you, as well as block, where you can use a system where you can directly block proliferation in the cells. And I'll guide you through a bit the mechanism that we have used. So let me guide you through. What you're seeing on the left is a cartoon where, again, these growth are made of red and green cells, so mutant and wild type. And what you're seeing here is the equivalent growth as it looks in real life, and this dash. And what you're seeing here is this hot dog-like shape where the mutant cells are in the middle and these wild-type cells are surrounding it. So when Samara and Christy blocked Wnt's feedback loop by using a Wintless loss of function in the same cells that also are expressing the mutation, they block this feedback loop of Wnt's from the mutant cells into the wild-type cells. And what you're seeing is that these mutant cells now are sticking out and there is less wild-type cells surrounding it. So they decided to push it further and ask to block instead proliferation in all these wild-type cells. And the genetics is a bit more complicated, for which they had to have two parallel systems, a Cre system for the mutation and a TET system for the block of proliferation. And I'm happy to answer questions for that. But if you just follow the picture, what you're seeing is that when we block proliferation more prevalently in these wild-type cells in green, what you're seeing now is a much larger growth, indicated here in red, and not green cells surrounding it. And when we tested the regression over time, at the same time window where a just mutation allows for 80% rate of regression, with the wintless full knockout, you have 40%, and with blocking proliferation, 2%. So this not only shows that there is less recruitment of these wild-type cells around it, but it has a functional role in which it will block regression. Okay, what does it all mean? At the tissue level, I don't think we had imagined that the epithelium per se could be capable of having ways of recognize and correct and go back to homeostasis if it wasn't, for instance, for recruitment of immune cells. That might still be happening, but the fact that the epithelium per se as a system that can allow to understand what's the blueprint to return to felt fascinating to us because we are speaking about adult homeostasis. It's not a developmental program of changes. The other thing is that Tianqi and Christy and Sarah went on also to understand there are mechanisms of prevention, not only correction, because we extended our understanding to additional mutation like HRAS and KRAS. And finally, that there are mechanisms for which we had not anticipated, not only at the signaling level that I won't have time to talk about, but also as the homeostatic principle of regeneration of the skin, proliferation per se. That in itself is a way to correct and prevent and allow an intrinsic defense mechanism that the skin epithelium is using when interfacing with oncogenic mutation. So in this part of the lab, we are very excited to continue with the other dimension, Anupam Malafa, Tianqi Qin, and Ingrid Hoyman, where they are respectively expanding our understanding of metabolism. Anupama's work was presented in a subsection of the Cancer Center with Rachel Perry, who is also part of the Cancer Center, and we team up together to understand metabolic rewiring. Tianqi has ventured and pushed the understanding from ERK sensor signaling dynamics all downstream of HRAS and KRAS. And Ingrid is bringing a cell geometry side to the understanding of how an healthy tissue interfaces with oncogenic mutation. In addition to the oncogenic mutation, another side of the lab is very interested in expanding our understanding of tissue types. So we have been focusing on the stem cell layer and the epithelium. There are all these other tissue types, fibroblasts and vasculature, they are nearby, that we started to explore over time. And so understanding how the niche or different tissue types regulate epithelial regeneration has been another area of research. In particular, something that we have been very curious has been the interaction between fibroblasts and epidermal stem cells because of large literature that shows the impact that the fibroblasts have. For instance, work from Howard Green has shown that when you take epithelial stem cells and you put them in culture, if they don't have fibroblasts, they cannot proliferate, and they cannot form a proper architecture that then is used right now in the hospital to transplant and repair wounds by burns. So the question we had is, what do we understand of this relationship in vivo, as this question has not yet been addressed when looking at the skin epidermis. And so the question that Bella and Shanshan are interested about is how fibroblasts impact epidermal maturation and maintenance. And the reason why they've been interested to do that in collaboration with Deandra Simpson is because we have an opportunity of understanding how the principle of regeneration are first established and then maintained by leveraging early stages that we had not explored before, such as neonate stage and all the way into adulthood. So the way that Bella and Shanshan decided to do that is by using ablation approaches. So if you have a population of fibroblasts and you reduce it by, again, a system that is Cre-mediated, which means restricted in spatial and restricted in time, what are the consequences for epidermal architecture and stem cell proliferation? And so by starting in neonate, what they did is to start to image again live mice that are anesthetized, in this case neonatal around P10, and then understanding how these fibroblasts underneath the epithelium I've shown you are distributed. In this case, it's very interesting to shift the tissue because you might not even realize which one is a cell because it appears as a continuum. But upon ablation, what they saw is a reduction of the coverage in this area. And what is interesting to me is that if you flip the way you look at them and now you look with nuclei so that you can be sure which one is a cell, the reduction we see from a control to a depletion is much greater than the reduction you are seeing here for the coverage. To us, this tells us about the homeostatic principle by which this population needs to come together and create much more of a continuum, even when they have a depletion in number of cells. So this depletion were all driven by a promoter that is expressed both in fibroblasts, but also adipocytes. So we have used areas in which adipocytes are less enriched. And this is a recombinable diphtheria toxin that when expressed is gonna kill the cells. And so in doing that, and then Bell and Chanchan wanted to understand that as we look at these fibroblasts and we play with their number and we reduce it, what we understand after this reduction about the impact that this dermal layer may have on tissue architecture and stem cell proliferation. And I think the most striking result is that there was no impact on the epithelial stem cell layer, especially proliferation. And that was really interesting to us. So if you take left to right, what you're seeing is a top-down view of the basal stem cell layers, where each nuclei is now colored with DAPI in magenta. And then we can use assays such as, for instance, phosphoate3 that tells you which cells are proliferating in that moment. And then we can do quantitative analysis to understand differences. And what we saw is that there was not a change in proliferation where we instead expected a difference in proliferation, but there was a change in density. What you're seeing here is the same channel that before was in magenta, now showing you simply the nuclei. And so what I told you then is that this epidermal, by reducing this fibroblast, this layer has an increase in density that happens despite proliferation being unchanged. And so what Bell and Chanchan then reason is that perhaps the process I told you at the beginning of the talk of departure from this basal layer to form these differentiated layers may be altered. And so that this delamination may be the problem. And so as you know, we can track the same tissue over time, but we can also do treatment like incorporation of EDU in the DNA, and then see upon chasing whether or not the cells are staying in this basal layer or leaving and progressing. This is through EDU labeling. So let me guide you through this assay. So what you're seeing here on the left is all the nuclei that in the basal layer and the upper layer are positive for this EDU. But what you're seeing is multiple layers, so different spatial location now flattened into one. And you've been shown the position by this color approach. So if you're still in the stem cell layer, you're more into the red and the pink. If you are going to the upper and differentiated layers, you are in this cyan colors. And so now in one inset, in one projection, you can compare your control to your manipulation and ask where are the cells in the depleted fibroblast. And what you see is that there is much more red propensity than cyan, indicating that the cells are more in the basal layers and have not departed at the same rate. So what I told you so far is that when comparing control to fibroblast reduction, that we are seeing a not change in proliferation, but this higher density. And that allows for Bell and Chanchan to start to ask, how is this departure and this higher density may be regulated? And since they are sitting on this basement membrane, they wanted to ask whether the basement membrane is affecting either composition and or function. So we started with a collaboration with Sarah Wikström and Clementine Villeneuve. And what you're seeing here is their analysis through EM, so much broader approach to ask what are characteristics that show up by EM when we compare control to fibroblast depletion. And in addition to see that there was a thickness increase in those spacement, there was also other property that we are going after now, including waviness, that might speak about a different way in which these basal cells are sitting on that basal layer. So this prompted Bella and Chanchan and Deandra to ask, okay, can we go molecularly in characterizing that basement membrane? And we know that these two components, laminin 511 and collagen 4, are secreted by the fibroblast. So we expected that we would see a reduction in those components. So what you're seeing here in the control is the red and green representing these two components. They compared to the fibroblast reduction show a different distribution from the one we had imagined. So in this case, we are seeing collagen 4 distribution that is diminished here along this epidermal to dermal junction. But what Deandra saw is that laminin 511 was much more upregulated in the depletion. So that was interesting to us because there were data that show in the past that laminin 511 is actually softer, providing softer properties. So Walch and Tran wanted to ask what are the basement membrane mechanical properties, and teaming up with Sarah and Clementine did atomic force microscopy that showed that in fact there is a reduction of stiffness in this basement membrane. And so altogether, this seems to reconcile with the fact that the property of laminin is softer, and with property that may be the cause of which this departure from the basal layer is being delayed. So what I told you for all this part is that we are very interested in trying to have a full picture of the skin. So not only epidermal regeneration by looking at epithelial principle, but also understanding how nearby tissues such as the fibroblasts and the vasculature play a role into this regeneration or not. And so when starting with the neonate and establishing this principle, what we realize is that there is an increased density in the stem cell compartment where fibroblasts do play a role for that, as well as changes in basement membrane mechanics and composition. But interestingly, when we follow this over time, we saw that in adult, there was actually no change. And so this prompted us to be curious about how time and age may influence these processes, and create a curiosity also about much later stages and what would happen in aging. So I'll leave you with the last piece of data, because we have been interested in aging in the past with a project by Edward Marsh, where what it was interesting is in understanding how this fibroblast distribution may evolve over time. And so what you are seeing here is a large field of view, again in a live mouse, where each individual dot is the nucleus of a fibroblast, and you're seeing this dense distribution of these cells. But when you will go over time, for instance, in older mice at 13 months, you started to see that there was loss of these fibroblasts in clusters. This was not something we expected, because we had imagined that, as we know in human, that you can lose fibroblasts, they would be much more equally distributed throughout the tissue. And so this cluster loss was really exciting to us. And what Edward decided to do is to revisit exactly the same area, the same cluster, to discover that these clusters should remain in the same place, but the new clusters would also form. So he opens at all type of questions of why are these clusters formed? Is it a cell that dies and then neighboring are dying because of toxic release? What is upstream of that, and what are downstream consequences? And it reminded us work done by Chen Kam, they also longitudinally tracking the same vasculature, saw this falling apart of the vasculature, where in the same mice, the same loops look much more tortuous and aberrant in the same mice. And so in the lab, we are moving to try to understand triangular regulation that are also molecularly supported for understanding how stem cells, fibroblasts, and vasculature may be having a relationship that is functional with each other or independent, and how this could evolve over time, as well as integrating it with the ever-evolving genetic moiseism that we know is happening in the epithelium, and asking questions, for instance, how does a decay niche impact somatic stem cell regeneration? And with that, I'd like to thank very much the collective lens that each individual member in the lab provides to. I'm highly indebted by two spectacular scientists, Dave Gonzalez and Kat Martone. We've been together for 10 years, and it has been really an adventure to continue to grow thanks to them. Tianqi Qin, an incredible pillar in the lab, is a fantastic scientist on faculty job right now. Former lab members that have gone on to a number of different professional and the war, industry, academic, teaching, and also hospital. And collaborators and teachers, not only with respect to research, but also for my understanding of my own place in the world, and anti-racism courses and mentoring, as well as someone that I bring within my heart and my behavior, always trying to uphold to the principle by which she led their lab, and the funding without which we couldn't do the research I presented. Thank you.
This was really cool, the vertical expression that you were seeing using the MF2 system. I was curious, what's the period? I was trying to get a concept of the periodicity.
Yeah. Jason, Megan is asking the periodicity of K10 or K14 of the burst.
Yeah,
No, the frequency.
Oh, the birds?
Yeah.
Oh, it's really, it varies from access down, like, page. Yeah, you can see that.
Jason is comparing both a differentiation as well as stemness gene, and then Han is doing actin as a housekeeping genes. So between these three, we hope to have a deeper understanding of that.
So there must be an active process for those cells to die, and how do the wild type cells?
Yeah. So let me frame it first that what we notice in these precancerous stages and benign tumor, that the processes leverage the homeostatic processes in place. There is no death. There is proliferation and differentiation. But what happens is the stem cells, which are wild type predominantly, occupy the basal layer on top of the basement membrane and allow for the mutant cells, so the mutant cells have a predilection to differentiate. So the elimination is by differentiation. You create this differentiation structure that then are extruded from the tissue by leveraging homeostatic mechanism.
So they have the benefit of the fact that you're always differentiating cells.
Exactly. So principle of cell competition in Drosophila, Hines Morata, and many others had shown apoptosis is certainly a mechanism. But I do imagine that the competition and collaboration mechanism must be in negotiation with what homeostatic principle normally the tissue is using. And apoptosis is not one of them in the epidermis. That's right. The mutant cells get initially, that's why we could do the trick of blocking proliferation, because those cells, the mutant one, are more likely to differentiate than proliferate. And instead, the large majority of proliferation was happening in the wild type cells. And so that's why those genetic mechanisms worked, blocking proliferation mostly in the wild type cells.
Have you looked at, or you want to comment on regional differences in skin, like here versus trunk? It looked like a lot of what you were looking at later on was interfollicular as opposed to follicular.
Absolutely. Yeah, absolutely. So geographic is important to us. The back is possible to image, but then you have to induce an injury. And so the ear where we image is the closest to the back, but there is always the question of what may be specific and what is not. Publications have shown distribution of collagen and type of collagen be different and therefore influencing. The tail is also another fascinating geographic region where there is separation of different stem cell types depending on the scale and interscale. So yes, geographic region does matter, of course. Thank you. Oh, on the chat. Let's see if I can. Megan, I cannot find the chat. Oh, in the top, I can. Oh, yeah. Okay. Record your attention to record this, okay? Yeah, I got it. Dr. Greco, thank you for this excellent talk and for your comments about passion and privilege in the beginning. Do you have suggestions about how to build a supportive and diverse lab? Suggestions about how we can build a supportive and cancer science? Yeah, thank you so much, Dr. Alpert, for the appreciation and the space to speak about that. I think more pressure on us in leadership must be done by the system and ourselves in order to really understand where we are going wrong. Because, for instance, I've been participating to forums, fire chats in other departments, and each department has a vice chair of diversity, each department has a council, and yet I still hear voices saying, I don't feel safe to bring up an issue, who should I go to? I think that tells us that the approaches we are taking are failing, and probably because we are not committed to really have a deep change. And so for me, it's been a wrestling, where do I profit and benefit from the system as it is before I can really move into being meaningful. In the department, we have taken an anti-racist course, for instance, that has allowed us to be in conversation. The same quotes I showed you, which cost me trepidation, and I actually was speaking with members in my lab to gain the courage to, are of course things happen in our home. And so unless we have spaces where we can talk across groups, we are never going to have a meaningful change, in my opinion. So both be demanded from the top and our own responsibility to continue to work to be better. Yeah, and I think in the cancer center seems to want to do something, seems to have resources. And so I think continuing to be in conversation. One thing I have, for instance, I ask in my department is to have somebody who has a professional role and the competence and the scholarship about the internalized inferiority with which we look at each other, whether it's a power construct of racism, a power construct of gender, you name it. I think unless we bring the expertise, what we're going to do is continue to reinforce this system and perhaps even be looking good on the way, but not changing. And I have to say that while I put up those episodes, I can tell you that the stories I hear in my role as a co-chair for SWIM are atrocious and tremendous. And we continue to do damage to each other. So I think there is a need for intervention. And can audience question please be repeated? Did I repeat the question? I didn't, sorry. Messed that up. Hey, Michael, so nice seeing you.
Clusters lost. I guess one was there were also single cells that were being lost, but the pattern is sort of shown. These definitely losing big chunks. Do you have an hypothesis as to why? Is it cell-cell interaction based?
Yeah, so the question has been, how are these cells lost in clustered? Is it a cell-cell interaction? These are a great question. Right now, we don't have the resolution. Those were 3 months apart. So it would be really important to have that type of resolution and understanding, is it a chain reaction? You start with one, but also understanding what is upstream. For instance, Edward started to zap some vasculature to see whether as a ripple effect, then you will lose fibroblasts nearby. So there is a lot to ask that, and it's a great question. I wonder if also senescent reporters could start to give some clues of why some cells may be lost, that then create an environment that is not viable for others. Yes.
Wonderful talk.
Thank you.
I think that the calcium signaling is really exciting to see, and interesting that it was in individual cells.
Mm-hmm.
I wonder if you have some ideas and started to look into like what's triggering it. So stochastically almost, it seems.
Yeah, it's a great question. So when one way to go about it, which is not precise, is first of all cataloging, right? And there are few ways, a lot that are single, and a consistent portion that was neighborhood between 2 and 10. And so we began there to try to understand those. But those single cells were also thinking, are they signaling center or repeated signal over time? The best answer we could do is just decided to go over time and ask whether those are persistent signaling or they change. And so in terms of role, who is single versus neighborhood, we realized that over 24 hours, everybody is competent to signal. These questions, I think, are really important and now being translated by Anusha Manitan in the lab, as well as Chen Kam for understanding vasculature, calcium in the vasculature. And there, instead, the principle seems different. There are some conservation in the way that we see those patterns happening, not only a day later, but also 14 days later. So where are these differences? What is upstream? Is this system for the tissue to say it's just a, saying we are alive, we are ready to signal? Is this something functional? All these questions are still up in the air.